Hi, I'm presenting the work Predicting Protein Secondary Structure Through Nearest Neighbor Search and Method Hybridization by Spencer Krieger and John Kececioglu. I'm Spencer Krieger and I'm excited to share this work with you today. Protein secondary structure prediction is a classic problem in bioinformatics. Proteins have four levels of structure: the primary amino acid sequence, the local and discrete secondary structure, the global tertiary structure, and the quaternary structure, or how protein modules interact. For secondary structure, given an amino acid sequence, each amino acid is assigned one of 3 or 8 secondary structure classes, depending on the model. The task is, given amino acid sequence P, predict the corresponding secondary structure sequence S of the folded protein. Our new tool nnessy is an algorithmic approach to protein secondary structure prediction, which avoids expensive SciBlast database homology searches. nnessy is a template-based method and uses k-nearest neighbor search over a smaller template database of fixed-length protein words with known secondary structure. These words are searched under a distance metric that obeys the triangle inequality, so we can leverage algorithms for nearest neighbor search over metric space, so the problem is much more structured. nnessy also computes a maximum likelihood physically valid prediction via dynamic programming, where the dynamic programming satisfies minimum run lengths, even for 8 states, runs in linear time, and is easily adaptable to other tools. Instead of outputting confidences, nnessy uses a feature-based accuracy estimator that takes a feature of the prediction process and estimates the unknown true accuracy of its prediction. We combine our template-based tool nnessy with a template-free tool to yield a hybrid method that gains the advantages of both tools. This hybrid method exceeds the state-of-the-art accuracy by more than 3% for the 3-state model of prediction and more than 7% for 8-state prediction. Next, I'll give an overview of nnessy's prediction process. Given an input amino acid sequence and our preprocessed template database, we perform nearest neighbor search on words from the input sequence to find their nearest neighbors from the template database. These nearest neighbors are given to an overlapping words procedure that combines the secondary structure sequences from the template words to estimate structure state probabilities at residues. These state probabilities are fed into a dynamic programming algorithm that does not output a greedy prediction that simply chooses the state with highest probability, but instead computes the physically valid structure of maximum likelihood. This maximum likelihood prediction is output by nnessy. To get state residue probabilities, we use the following procedure. Given an input protein sequence, we extract fixed-length words at every position, where each colored box here represents a different fixed-length word. To obtain state probabilities for this residue highlighted in red, for each word that contains this residue, nearest neighbors are found from our template database using the dispersion tree data structure. These template words contribute to the state probabilities of each residue they cover, where the structure states are indicated by the colors of the letters, where alpha helix is shown in blue, beta strand is shown in green, and coil is shown in black. So for the highlighted residue, the frequencies of structure states of the template words at the box positions determine its state probabilities. For this example, the frequencies of alpha, beta, and coil give the structure probabilities 0, 0.62, and 0.38. These state probabilities are then used in a maximum likelihood prediction procedure. The procedure considers physically valid secondary structure sequences that satisfy minimum run length constraints on structure classes. We find the optimal physically valid structure sequence of maximum likelihood by dynamic programming. The maximum likelihood objective function that this algorithm optimizes is the following product, which has factors for state probabilities, transition probabilities of one state to another, and run length probabilities. We minimize the log likelihood of this objective function by solving a recurrence equation. The dynamic programming algorithm computes a quantity L of ILC, which is the negative log likelihood of an optimal prediction ending at position I with structure class C and run length L. Below, we give one of 3 cases, the general case of L greater than minimum run length of class C. Here, L of ILC is a sum of state, transition, and run length probabilities, and the negative log likelihood of an optimal prediction at the previous position. Given a sequence of length n, the dynamic programming algorithm runs in linear time. A unique aspect of our approach is that it uses an accuracy estimator, which estimates the unknown true accuracy of its prediction based on an easily computable feature, which is average word distance of overlapping nearest neighbors in our case. Each circle in this figure represents a residue from Cas proteins, which is plotted at its average word distance on the horizontal axis. Its value on the vertical axis is average empirical accuracy of residues with similar word distance, which is averaged over the protein to get protein estimated accuracy. The dashed lines give the piecewise linear fitted estimator, which maps word distance to estimated residue accuracy. The resulting accuracy estimator has an average error of 5% for the 3-state model and 8% for the 8-state model. This hybrid approach combines nnessy, a template-based method, with a template-free method. Given nnessy template-based prediction and the prediction of a template-free method, we estimate the accuracy of nnessy template-based prediction. If the estimated accuracy is above a threshold, the hybrid outputs nnessy prediction. Otherwise, it outputs the alternate prediction of a template-free tool. The hybrid gains the advantages of a template-based method, where when a good template is found, the prediction has high accuracy. We combine nnessy with a robust and stable template-free method, so when no good template is found, the accuracy of the hybrid prediction is robust and stable. We now give the prediction accuracies for state-of-the-art tools including nnessy and the hybrid method. These tables give accuracies for the standard 3-state and 8-state models of secondary structure prediction. We use Q3 or Q8 accuracy, which is the fraction of residues with correctly predicted secondary structure. The hybrid boost is the increase in accuracy when hybridizing a given tool with nnessy. The best nnessy hybrid is 3% better than any single tool for 3-state and 7% better than any single tool for 8-state prediction. Thank you for listening to my presentation.